Hello, everyone. Good afternoon, everyone. I'm one teacher. I'm Mrs. Wang Huang. I'm working in Wenzhou Medical University and teaching pharmacology. So today we'll talk about some traditional Chinese medicine. The first one we'll talk about is one of the first Chinese medicines to be imported to the West from China. It is Da Huang, in Chinese, Da Huang. It is Da Huang, and English name rhubarb. That name reacts at rhizomazine. So these cool drugs are rhubarb. Rhubarb roots have been used as laxative for several millennia. Rhubarb also appears in medieval Arabic and European prescriptions. We introduce this cool drug as following contents: source, biological source, plant morphology, macroscopic characters, microscopic characters, or microscopic identification, constituents, physical, chemical identification, and the effects. So every cool drug we'll talk about is as the following contents. So first, source, biological source. Usually, sometimes we call rhubarb as call it general or 川菌. Why do we call it general? Rhubarb is known as a general because of its powerful effect. So it is called general. It was first recorded in the Sinon Benchao Classic. Biological source. Rhubarb is a dry root and rhizome of 3 species. They are rheum permanentum, rheum tanguticum, rheum officinalis. These 3 species, root and rhizome, used as rhubarb cool drug and in family Paniculaceae. Now let's introduce plant morphology. The common characters of Paniculaceae in this family having osseate, simple arranged flower, stamen 6 to 9. But different plants have different morphology. So respective characters of the plant in rhubarb, rheum permanentum, have the leaves palmatifade, flower purple. Rheum tanguticum, the leaf is palmately parted, flower purple. Rheum officinalis, the leaf is palmately beige and the flower is yellow. Now let's look at the pictures. The left picture is rheum permanentum. So we can see from this picture, the leaf of this plant, the leaf is palmatifade, and the right picture, it's rheum tanguticum. The leaf is permanently parted permanently parted. Room first nail, the flower is yellow and the leaf is permanently beige. So these three species have their respective morphology. So the differences are focused on leaves and flowers. So how do we connect and prepare the crude drug? Usually we collect it in late autumn when stem and leaves are withered, or in next spring just before budding. And remove from rootlet and the outer bark, cut it into sections and dry it directly in the sun. So this is a collection of preparation. So microscopic characters. Usually when we talk about microscopic characters, there are some items we should introduce. One is shape, size, color, texture, feature, and odor and taste. So first, rhubarb's shape. The shape is subcylindrical, conical, hoof shape, or irregular shape. So look at this picture. The left, the shape of the crude drug, cylindrical or irregular black. So this is the shape. Some of them are conical or hoof shape. So this is the shape of rhubarb. Size 3 to 10 centimeters in diameter. Color. What's the color? Brownish yellow to brownish yellow or reddish yellow or yellowish brown or reddish brown. So this is the color, and the texture form is heavy. Yeah. Texture form and piece broad. So here the piece, this part so this part belong to a piece. These are longitudinal section, and the right picture are transverse section. The transverse section, we can see this part belong to this part is a piece. There are some star spots in the piece. They are arranged in ring or irregular arranged. So where is star spots? Here. Yeah, star spots. Yeah, star spot. Here are star spots. So these are star spots. They are arranged in rings or irregular. So in the piece, there are so many star spots. The piece is broad. So these are transverse sections. Picture. Odor, taste, aroma. Oh, sorry, smell, aroma, and taste bitter and slightly astringent, sticky and gritty on chewing. So these are microscopic characters. When we identify the crude drug, the microscopic characters was one of the methods to identify microscopic characters. So these are room, permentum, rhizome, and root. Zoom of snail. The left picture is zoom for snails transverse section. Also, there are some star spots in peace. So the organ is rhizome. So the right picture is room tanguticum. Also, we can see some star spots. So these are star spots in it, in peace. These are in peace. So these are microscopic characters. Next one, microscopic characters. The picture shows us the transverse section of rhizome. From outside to inside, the outside, this part belongs to cork. It is cork. Cork contains several layer cells. But most of the cork removed. This part belongs to cortex. And this part, from this part, they are phloem and the cambium, and this part is xylem. And peace, the center is peace. Here are some abnormal vascular and phloem and xylem. So these are abnormal vascular in peace. Pittsburgh usually showing abnormal vascular bundles scattered and the parenchymal cells containing abundant calcium oxalate receptors or clusters. So these are microscopic characters. Microscopic characters of the powder of the rhubarb. We grinded the food drug into the powder and make a temporary slices and put it under the microscope to observe. There's some tissue we can see clusters. So the first one is clusters. The clusters abundant pyramids, obtuse. And starch grains, here are starch grains, starch granules, single compound, and vessels. 3 is vessels, bordered, pitted, reticulate, spiral, and annular vessels, 11 to 140 micro in diameter. So these are powders, microscopic characters. Room permentum, upper picture. The figure clusters. So like here, this is the calcium oxalate cluster. So these clusters is subcycloidal. The petals of cluster are more, edges and collars are mostly short and small without symmetry. Compound starches, these are compound starches. granules are consisted by 2 to 7 branch granules. So these are vessels. The lower picture. These microscopic characters is coming from zoomer Fresnel's microscopic characters. The figure of the clusters is subcolloidal. The edges and colors are mostly broad and symmetrical. So these clusters, the edges, the edges and colors are mostly broad and symmetrical. Compound starch granules are consisted by 2 to 9 branch granules and some vessels. So these are microscopic characters of the powder. It contains mainly 3 kind of microscopic characters. One is clusters, 2 is vessels, and 3 is compound starch granules. So these are microscopic characters. Next one, constituents. A medical analysis indicates that rhubarb contains endocrino derivatives. It includes free endocrinos and combined endocrinos. Free endocrino, such as amylin, crisophenol, and amylin, ferritin, etc. Hello? Ferritin, etc. So this free endocrino has antibacterial effect. Combined endocrinos, such as glucosides or diasterones of the free endocrinos, such as sinucide A, B, C, and D. These combined endocrinos usually used as diuretic constituents. So the constituents of the rhubarb include endocrinos. It includes free endocrinos and combined endocrinos. Usually free endocrinos has antibacterial effect, and combined endocrinos are the diuretic constituents. So these are constituents. So these are structure of free endocrino and combined endocrino. So different group here is R group and R1 group. So different group, different constituents. So here, constituents are compared among these 3 species. It includes endocrinos, total endocrinos, combined endocrinos, and free endocrinos. So in 3 species, the table indicates that the free endocrino in Zoomer Fresnel, here, is the lowest. So this is a comparison of constituents in 3 species. Next one, physical chemical identification. Usually we identify this drug by physical or chemical identification. It includes such as microsublimation and fluorescence tests, tests for combined endocrinos and TLC, etc. Here, microsublimation test indicate the result. The yellow needles or feather-shaped crystals exist, but can be observed under the microscope. So the yellow needles, like this picture, in this picture, the yellow needle shape. So these crystals is connected in lower temperature. High temperature is feather-shaped crystals. So these feather-shaped crystals. So these are micro-sublimations. Fluorescence test. Take some powder into a test tube and some dilute alcohol, and then filter it. Take 1 to 3 drops of filtrate on a piece of filter paper and dry it, and then put it under the UV light. The cool drug shows brown-red. It indicates this cool drug is official varieties. If it shows bluish purple, it indicates it's fake. So this is a fluorescence test. The color is different. The official varieties shows brown, red, but fake shows bluish purple. So this cool drug can be identified by fluorescence test. So let's test for combined endocrinos. So this is another test, biological test. So here we don't talk about in detail, and the result, red color. If it shows red color, it's official rhubarb. So and a TLC. So this is a test solution, reference drug solution, and the reference solution, and the plate, mobile phase. The result, examine under ultraviolet light the 5 orange fluorescent spots in the chromatograph obtained with the test solution correspond in position and color to the spots in the chromatograph obtained with the reference drug solution. Spot becomes red. Becomes red under sunlight exposure to amyl vapor. So here this is the result. Result, TLC result. So these are about chemical identification. Sometimes some adulterants are misused. These are adulterants, means they are fakes, like room emery, ruhotausins, etc., room nobile, and room chanipensis. So these are adulterants, not a genuine rhubarb, although it came from same family, even same genus, but they are not really rhubarb, they are adulterants. How do we identify these adulterants? Here, it can be identified by fluorescence tests. put it under the UV light. The transverse section shows the bluish-purple fluorescence in the ultralight. So there's a bluish-purple. So it indicates this cool drug is fake, not genuine rhubarb. Genuine rhubarb shows brown. So it's another adulterant. The point of identification: thin and small texture form but light, and no star spots. Yeah, star spots, we know that the microscopic characters, there are so many star spots in pith, but in this adulterant, there's no star spots. So it indicates this cool drug is fake, not genuine rhubarb. So next one. This adulterant, look at the picture. Where is the pith? Here, it is the pith. Is there any star spots in it? No, so no star spots. So it also indicates this cool drug is fake and not genuine rhubarb. It's fake, adulterant. Next one. So this cool drug also indicates there's no star spot in pith. So it's also fake. Next one. So sometimes somebody misuse this and rhubarb. It looks like rhubarb, but it isn't rhubarb. Usually, the microscopic characters show there's no star spots in pith, so there is not any star spots in pith. It indicates this cool drug also fake. Next one. So the point of identification also is microscopic characters and radio green, but no star spots. And Nobel. Look at this picture here. So the piece has no star spots. Also indicated this cool drug is fake. So this one, no star spots in piece. Now from the transverse section here, transverse section, there's no star spots in it. So also it indicated this cool drug is also fake. So how to identify genuine rhubarb and fakes? By the way, star spots in pith, but pith is listed in rhizome. Does root have pith? Remember, most root of the plant have low pith, so there's no star spots in only rhubarb's root, but rhizome different. So rhizomes, because there's some the piece in rhizome, so rhizome has star spores, but this is in rhubarb. So now, how to identify genuine rhubarb and fakes? Here's a summary. So difference between artificial varieties and fakes. Artificial varieties and fakes So these are comparison species. To look at looking for star spots in rhizome, not in root. Remember, not in root, in rhizome. So the piece, star spots in piece. Official varieties having star spots, but figs have no star spots. So this is your first difference. And odor and taste. Official varieties smell aromatic. Can you hear?
Yes, ma'am.
Oh, okay. Official varieties smell aromatical, but figs smell odorless. Official varieties taste slightly astringent, but figs also taste slightly bitter. These are microscopic characters. Also, we can identify the cool drug by fluorescence test. So official varieties put this cool drug under the UV light, and the cool drug, if shows brown to reddish brown, it indicates these are official varieties. And figs shows bluish purple. So these are the main two differences between official varieties and figs. And official varieties, the microscopic characters are different from figs, and the fluorescence tests also are different from the figs. So these are main method to identify genuine rhubarb or official varieties and the figs. So from the point of traditional Chinese medicine, rhubarb have the effect of clearing heat, purging fire, cooling blood, and detoxifying by removing accumulation with purgation. So there are some traditional Chinese medicine. So these are 君治牛黄解毒片 in Chinese name, and 麻仁丸, another traditional Chinese medicine. These traditional Chinese medicines contain rhubarb. So about the rhubarb, we are talking about mainly source, microscopic characters, constituents, and physical and chemical identification and its effect. This is all about rhubarb. So here, microscopic characters, the important characters is star spots in peace, in rhizome, and the microscopic characters include clusters, calcium clusters, starch granules, and vessels. And the constituents include anthraquinones. It includes the free anthraquinones and the combined anthraquinones. Free anthraquinones has antibacterial effect, and combined anthraquinones are negative constituents. So physical and chemical identification, it includes microsublimation and the fluorescence and other. So these are rhubarb. Okay, let's talk about other traditional Chinese medicine. So the next one we'll talk about is Shanja. Is this? So the Chinese name Shanja and Chinese pinyin Shanja are cretaceous fractures. It's a net name, cretaceous fractures, and whole thin fruit, English name, whole thin fruit. So about this cool drug, so this is a cool drug. The left picture is cool drug, and the right picture is the photo on tree. So these are introduction. Cretaceous, commonly called Hothin, is a well-known herb in traditional Chinese medicine and is an herb remedy in cultures throughout much of the world. Well, there is a temperate climate supporting growth of the trees, especially in Europe. The species used in China, Cretaceous pinnatifida, differs from that which is relied upon and the source of the Western herb, obtained mainly from Cretaceous margina and Cretaceous acantha. Western herbalists primarily use Hothin for treatment of heart disease. In ancient times, the Chinese herbalists mainly used Cretaceous to improve digestion. Numerous Chinese formulations now incorporate the herb for strengthening heart function, lowering blood lipids, and dilating blood vessels to promote blood circulation. So now we'll introduce biological origin. So the cool drug is the dry ripe fruit of the deciduous shrub or small arbor plant, Cretaceous pinnatifida or Cretaceous pinnatifida BG. So the left picture, look at the left picture. So this plant is a variant of the right picture, the right plant. So the left plant. This plant is a variant. So these two species ripe fruit are used as cool drug. So microscopic characters, the fruit. So just we talk about the microscopic characters from which items or from which content. So the first one is shape and size and color, texture, fracture, odor, taste. So let's introduce these cool drugs microscopic characters. The first, the shape. So what do you think the shape, the Hothin? So this fruit is spheric, 1 to 2.5 centimeter in diameter. The external surface of the fruit is deep red. jelly with tiny white spots. So it's here with tiny white spots is obvious, like the first picture here. So there are so many tiny white. So here, tiny white spots. Apical depression. So like here, apical depression with persistent calyx, faced with fine fruit stalk or stem scar, 5 seeds. So every fruit have 5 seeds. Seeds accurate, slightly fragrance, sour taste, slightly sweet. So these are microscopic characters. And persist usually dries fruit in the sun and cut into pieces, or unprocessed or stir-baked. It's simple. So constituents. Medical analysis indicates that porcine contain 2 kinds of active constituents. They are organic acids and flavonoids. So the 2 kinds of active constituents. Organic acids such as arsenic acid, onionic acid, citric acid, tartaric acid, and ascorbic acid. These organic acids are effective components of help digestion. Flavonoids such as quercetin, hyperoside, lutein. So these flavonoids are effective components to prevent cardiovascular disease and to lower blood lipids. So these are main 2 kinds of active constituents, organic acid and flavonoids. Usually flavonoids are effect on cardiovascular disease. So pharmacological effects. Pharmacological tests indicate that the porcine has following effects. One is cardiac function. Porcine has a role of increasing myocardial contractility, increase cardiac output, and heart rate slowing down. 2, uncolored blood flow and myocardial oxygen consumption. Porcine extract and flavonoids have increased coronary blood flow, reducing myocardial oxygen consumption and myocardial oxygen utilization rate. 3, on myocardial nutritional blood flow. Porcine on myocardial nutritional blood flow increase in a certain role. Porcine effect on myocardial nutritional blood flow may be associated with dilation of coronary vascular bed to improve coronary circulation related. 4, on experimental myocardial ischemia. Porcine extract has prevent or reduced myocardial ischemia. So these are pharmacological effects. From the point of the view of traditional Chinese medicine, porcine promote digestion and dissipate food stagnation, activate blood and resolve stasis. Usually, ascorbic acid improves the activity of proteolytical enzymes and help digestion, and lipids promotes lipolysis. So cardiotonic and hypertensive effects, the active components are flavonoids. at bacteria and at cancer effects. Also, we are the active components are flavonoids. So culinary use in the United Kingdom, they are sometimes used to make a jelly or homemade wine. In China, they are used to make many kinds of Chinese snacks, including whole flakes and tanghulu. They are also used to produce jams, jellies, juices, alcoholic beverages, and other drinks. So here, look at these pictures. These are, so the left picture, so these are whole flakes. So, and right up picture, these are tanghulu. Here's the tanghulu. So you generally, children like tanghulu very much. So these are jam and the juices. Also, it can be used to make poison tea. So these are tanghulu. So these are about, these are all about the horsing fruit. So, so next one. So now we talk about Huang Nian. So Huang Nian, this is Chinese name. So these are Huang Nian, Chinese name, and the Chinese pinyin Huang Nian, and the coopted is rhizoma. It's Latin name and English name cooptis root. Usually we call it cooptis. So these food drugs are cooptis. So we also introduce this food drug and the following contents: source, plant morphology, macroscopic characters, and microscopic characters, constituents, physical, chemical identification, and pharmacological effects and uses. So first, the biological origin. Cooptis is a rhizome of a perennial herb, Cooptis chinensis, Cooptis deotodia, and Cooptis tinda. So these three species rhizome. So here, please remember rhizome, not root, rhizome. So rhizome and the root are different organs. So you should know rhizome is a stem. Oh, sorry, it's a stem. It's a stem, but root is root. So they are different organs. So the medicinal part is rhizome, not root. The family Ranunculaceae. So here, plant morphology. The plant of Cooptis chinensis is a perennial herb. Root and rhizome is yellow. So from the middle picture, we can see that the root and rhizome, the color yellow, branched often and densely covered with many fibers. So here from the right picture, we know that root and rhizome are yellow and densely covered with many fibers. All leaves are basal, petiole. So petiole is from 5 to 16 centimeter. Blade, so here blade papery, ovate triangular. So it's the ovate triangular. So it's the leaf. Do you think so? So here, look at the middle picture. blade. So oval triangular, 10 centimeter wide and 3-lobed. Flower stems are 1 to 2 in number, equal or longer to leaf in length. Bracts are lanceolate and 3 to 5 pinnate, partite, scissor 7 to 8 pieces. Oblong, about 2 millimeter and broad. Its flowering period is from February to April and fruiting period from March to June. So these are Coptis plant morphology. So here is other species. So Coptis detail. So this is the kudjak, raw kudjak. And so this is the root and rhizoma. So here, this part is root, and this part, the big part. So this part is rhizoma. The rhizome is a medicinal part, but root is not. So here, Coptis theta. So here, rhizoma. So this part is rhizome, and here is root. So blade, oval triangular. So here are microscopic characters. Microscopic characters, sometimes we call it organoleptic characters. Why do we call it organoleptic characters? Because these features depend on our sense organ, such as our eyes, nose, and ears, and mouth, etc. So these microscopic characters, sometimes we call it organoleptic characters. So microscopic identification, also called organoleptic identification. So the first shape, what shape do you think? Rhizome, rhizome branch, the cluster, single rhizome cylindrical. So it's a single rhizome cylindrical. Size 2 to 7 centimeter long and 3 to 8 millimeter in diameter. Color, externally yellow-brown. And texture, relatively hard fracture. So here, fracture. Bark, bark dark brown. Where is bark? So outer part, this bark, this part belongs bark. So this part here, these are bark are dark brown. Xylem, bright yellow or golden yellow. Where is xylem? Here, this golden yellow, this is xylem. Oh, sorry. These are xylem. So here are xylem, and xylem. So xylem, bright yellow, golden yellow. Peas, red brown. Where is peas? Like here, this is peas, or the center of the drug is peas. So these are peas. The peas, red brown, sometimes hollow. So this is a fracture. Odor, smell odorless, and taste very bitter. It's very, very bitter. And in China, there is a Chinese saying that mute people eat Coptis and cannot speak out bitterness. It indicates that the Coptis is very, very bitter. So these are microscopic characters: shape, size, color, texture, fracture, odor, and taste. Sometimes we can identify the drug by microscopic characters or organoleptic characters. So next one, how about other species of coptes? So the upper picture is rhizome or Coptis deotodia. So Coptis deotodia's rhizome is single branch. Interlude. So where is interlude? Like here, it's interlude. So interlude longer, smooth. So here also it's interlude, longer and smooth. So in this picture, Coptis cheetah. Look at this picture, Coptis cheetah. Usually they are single branch, and interlude is not obvious. So it's not obvious, just not obvious. So there's a difference among the three species rhizome. We put the picture together to compare. So the first one, for this part, the upper picture, so it is Coptis chinensis rhizome. So we can see this rhizome usually mostly branched. Do you think so? This picture, it looks like what? I think it's like chicken feet. So like a chicken feet. So in Chinese is chicken feet Coptis. So it is branched. And the other two mostly single branched. So it's a branch. Interlude. So here interlude is obvious. Interlude, single one, the single one. Interlude is obvious and smooth and long. So here, Coptis deotodia, interlude is obvious and smooth, but Coptis cheetah, interlude not obvious. So these are microscopic characters. So three species, the main differences. Next one, microscopic characters. Microscopic characters include the transverse section and powders microscopic characters. So the first transverse section is microstructure. From outside to inner side, the microstructure includes the cork, the first layer, the cork. Cork consisting of several layers of brown cells. And the cortex, this part is cortex. This green part is cortex. Cortex is relatively broad. Stone cell, bright yellow, this is red. So this red part are a group of stone cells. So these stone cells distributed in cortex, a group of stone cells. And where is phloem? Here is phloem. Now phloem is very narrow. Phloem. Also, this part is here. This is a phloem. And cambium. Cambium is inner ring, so we call it cambium. And so this part is xylem. Flow of stone cells and Oh, sorry. So here, the central part here, central part is the piece. The piece contains. So these are xylem, so xylem, and the phloem. So this part is the phloem, and the phloem, near the phloem, there are group of stone cells. So these are microscopic characters of transverse section. So 3 species transverse sections, microscopic characters. So 1, 2, 3, 1 to 10. Every species contain 1 to 10 microstructure. So 1 here, cork, yeah, and stone cell. Red part is stone cell. These are stone cell. It's cortex. So and the phloem, cambium, and the xylem. And this rays, this part is rays, xylem rays, and the piece. So the middle picture also contains this microstructure. But what are differences among these 3 species? What are differences? Look at these 3 pictures carefully.
We not see your presentation.
What?
We cannot see the presentation.
Stone cell, look at stone cell. Stone cells.
We cannot see the presentation.
Yeah. Stone cell distributed in different parts. The first picture shows us then the stone cells distributed in cortex, right? So cortex and piece, there's no stone cell. The middle picture shows us that stone cells distributed in cortex, phloem, and xylem, and piece. So there are many stone cells, many groups of stone cells. But the next picture, the right picture, there's no stone cell, right? So stones in different species, these are main differences. So the first is Coptis chinensis. The middle picture is Coptis teotodia, and the next one is Coptis theta. So microscopic characters of the powder. The cool drug grinded into powder, and the makers temporarily and put it under the microscope to observe. Some tissues can be observed. For example, the stone cells, stone cells, and fibers too is fibers, bast fiber, and 3 is scale leaf epidermal cells. The powder's color here. The powder's color is brown and yellow. So the stone cells are bright yellow under the microscope. Channel distinct, a pit channel distinct. And bast fiber here, bast fiber are bright yellow, non-spindle or spindle shape, or thickness visible hole here. Hole is visible here, next is hole. it's visible. So these are bast fiber. These are bast fibers, and scale. V curved, beaded thicket. Beaded thicket means this silver, silver like this, like a bead, so like this. Like a beaded seal. So this is a silver. Look at here. Hello. Hi.
Hello.
Welcome back to our class. Sorry, the internet is not good. Okay. Can you hear?
Yes, ma'am.
Oh, okay. So continue. So these are microscopic characters. Pit the vessels or spiral vessels visible. So the main microscopic characters, three kind of characters. The first is the stone cell and the bast fiber and the scale leaf ectodermal cells. So these are microscopic characters. So next one, chemical constituents. Medical analysis indicates that corpus contain two kinds of. One is isoquinoline alkaloids. The other is manganese and ferric acid. Isoquinoline alkaloids such as berberine, colchicine, methylcolchicine. Do you heard?
Yes, but no screen.
Oh, okay. Okay.
No slides, ma'am.
Okay. Yes, not nice. So methylcolchicine and palmitin and glycyrrhizine, etc. So these are isoquinoline alkaloids. And fibers root containing berberine approximately 5%. Leaves containing berberine approximately 1.4% to 2.9%. So these are isoquinoline alkaloids. So manganese and ferric acid. So these are structure of.
Ma'am, there is no slide. We cannot see the presentation.
Isoquinoline alkaloids, different group, different and different isoquinoline alkaloids. We can identify the good drug by fluorescence test. Fluorescence tests indicate that xylem under the UV light, the color is golden yellow fluorescence. Yeah, golden yellow fluorescence. The berberine reaction. So take 1 gram of its fine powder, add 10 milliliters of methyl, heat until it is boiled in water bath, cool and filter it. Take 5 drops of hydrochloric acid and a small quantity of blanching powder and observe the color change. So this picture shows us the result of berberine reaction. The right one is extract, and the left one, this one is berberine reaction. So we can see the color changed from brown to red. So this is a berberine reaction. So if it is berberine, and screening alkaloids bubbling. So microscopic chemistry reaction. Take some powder on the glass slide and a drop of 70% ethanol and 1 drop of dilute hydrochloric acid and test the shape and color of the appeared crystals. So under the microscope to observe. So the numerous yellow needle-cluster crystals under the microscope can be observed. So what's these crystals? What are these crystals? These crystals are murate of bubbling, murate of the bubbling. So it indicates this food drug contain bubbling. So when you hit the glass slide and so dissolved. So this is a microscopic chemistry reaction. So pharmacological effects. Pharmacological tests indicate the properties has the effect of antibacterial, antiviral, and antiemetic and effect, and properties decoction, bubbling, and competition have broad spectrum antibacterial effect, anti-inflammatory and anti-ulcer effect, and hypertensive and paretical, gallbladder, artery, stimulant, and anti-cancer effects. So these are pharmacological effects. And uses, Copertis root is used to treat diarrhea and gastrointestinal problems, hypertension, and bacterial infections. Modern days, herbalists use the root for its antibacterial and antifungal properties, and still use it to treat stomach and liver disorders, pain, and infections. Berberine helps the following conditions, such as diabetes, heart disease, high cholesterol, high blood pressure, and immune challenges, intestinal infections, joint problems, noble density, etc. It can also help control weight. So these are Copertis uses. So actions from the point of view of Chinese medicines, Copertis can clear heat and dry staples. It is one of the strongest herbs to clear heat, dry staples, and eliminate toxins, treat traveler's diarrhea. So sedates fire, treat liver fire and heart fire, clears fire toxin, treat skin problems arising from toxic fire, applied topically. So here, please attention. This is a very cold natural problems from the point of view traditional Chinese medicine. Please do not use it for long time. So, so here are some compound Chinese patent medicine. So in Chinese, it's named 滋润烧伤膏. The main effect is to clear heat and detoxify, relieve pain, and promote muscle growth. Used for various burns, scalding, and burns. So I don't know if you can see me. So here, so
No.
so here, so I don't know if you can see this one. Can you see it?
No,
No.
doctor, we cannot.
You can see it.
No.
Oh, why?
I don't know, but it shows that we can see black screen.
You can see me.
No, doctor. Yes, now we can see the screen. Yes.
Yeah, yes, this one. Yeah. So like my hand, my here, so it's a burn. It's burned during the cooking. It's burned. So I use this traditional Chinese medicine to cool it, and so main effect is to clear, heat, and detoxify, relieve pain, and promote muscles. So used for various burns, body and burns. These are 滋润烧伤膏 compound Chinese traditional Chinese medicine, patent medicine. This medicine contains properties. So the effect is obvious. So other Chinese medicine like Huanglian, Sangqingwan, Sanhuangpian, these cool drug, traditional Chinese medicine contain properties. So they are compound Chinese patent medicines. So these are properties. So about properties, we talk about the source, macroscopic characters, microscopic characters, constituents, and the physiochemical identification, etc. So these are all about properties. Okay. Due to the time constraint, because of time constraints, we'll not introduce the properties in detail. And there are some other traditional Chinese medicine we haven't introduced. So, oh, sorry. Oh. Like ginseng, horsing, rhubarb, licorice. Maybe licorice in Russia. Do you know licorice?
No.
No, it's licorice. Yeah, because of time constraints, so we talk about this cool drug briefly. Not in detail. So the ganchao, Chinese name ganchao, in English means sweet herb. Yes, very, very sweet. So Chinese pinyin ganchao, and the English name licorice root. And radix glycerin, this is the name. So these are raw drug and slices. So the oranges, bare-knocked oranges. It's a dry root and rhizome of 3 species, family Leguminosae. So it is usually spelled licorice in British usage, but licorice in United States and Canada. So plant morphology, look at the picture. It is a perennial herb. And the stem, flower, and the leaf. So the leaf is compound, not single. So here we don't talk about, and we are this collect. Oh, collect and prepare the cool drug. And the microscopic chemicals, so this cool drug licorice and the slices of licorice. And microscopic chemicals, the microscopic chemicals of the powder. So it's crystal fibers, vessels, it's crystals. And the chemical constituent, here we should talk about chemical constituents. Licorice contains triterpenoid samplings, like glycerin, which upon hydrolysis yields glucuronic acid and glycerylic acid. So another food drug component is the flavonoids. So triterpenoid samplings and flavonoids. So there's a structure of triterpenoid samplings and the flavonoids structures. So pharmacology, it indicates that licorice has anti-ulcer effect. The drug is effective for treatment of gastric ulcer, probably because of the flavonoids it contains and adrenocortical hormone actions. And detoxify. So the detoxify effects are very important. Glycerin possesses detoxify action on drugs, poisonous food, and toxins of metabolic products. Glycerin, when degraded, yields glucuronic acid, which in the liver were combined with toxic materials to form glucuronide to produce the toxify action. So, and for agitative, expectorant, and anti-asthma effects, anti-inflammatory and anti-allergic effects. So from the point of view, traditional Chinese medicine, licorice is to reinforce the function of the spleen and replenish qi, to remove heat and counteract toxicity, to dispel phlegm and relieve cough, and to moderate drug actions. So these are indications. So it can be used in tobacco and food and candy and soil. And so these are tea and can be cut into slices to chew it. It's very sweet. So used in all kinds of snacks. So that's all about licorice. Okay. That's all for today's traditional Chinese medicine. So if you have any question, please send a message. So let's have a break.
Hello? Can you hear me?
Yes.
Yes, sir.
Okay. So when shall we start the class?
Yes, sir, we can start.
Nobody can hear you, sir.
Can you hear us? No, we cannot hear you, but we can see you. Your mic is off.
So now can you hear me?
Yes, we can hear.
So now you can hear me and also see me, right?
Yes.
Yes,
Okay,
right.
okay. Okay, now it's good because I re-entered the system. So now we start the class. So now I share the screen. Okay, so today I will talk about deubiquitinases as key regulators of neuroinflammation. So two things. First is deubiquitinase, and the second one is neuroinflammation. We have to link deubiquitinases with neuroinflammation. So what are deubiquitinases? And before at the beginning of this class, I want to first briefly introduce myself. My name is Xu Wang. I got my PhD degree at the University of Magdeburg in Germany. And after that, I worked in the University of Magdeburg for several years. Then in 2019, I came back to China, and then now I am a professor at Wenzhou Medical University. This is a picture of Magdeburg. In Magdeburg, Otto van Guericke did the experiment to show the existence of air pressure. And this picture shows the Magdeburg Cathedral. So, so we begin the class, and cells, they are living in a changing microenvironment, just like the human society, right? Our human society is very complicated, and the cells also have a very complicated microenvironment. This environment is fast changing, so the cells must react swiftly to the environment. Otherwise, they will have problems, just like people. So we also need to respond fast to the changing environment. Especially when there is infection, we know there are many pathogens, for example, viruses and bacteria. and the cells must react fast and correctly to the infections. Otherwise, the body will get a severe infection. So how can the cells respond to the changing environment? So when the cells are encountered by pro-inflammatory stimuli, for example, LPS. LPS is also called lipopolysaccharide, is a material from bacteria. And TNF is a cytokine. It's also, the full name is the tumor necrosis factor. It's a very strong inflammatory cytokine. When the cells are encountered by this pro-inflammatory stimuli, then the intracellular signaling pathways are activated. Of course, LPS and the TNF can activate the NF-κB signaling pathway. This signaling pathway is a very strong inflammatory pathway, and activation of this signaling pathway leads to the production of a lot of pro-inflammatory cytokines and chemokines. Then this will cause a proper inflammation, and this inflammation can kill the invading pathogens. But the signaling pathways, for example, this NF-κB signaling pathway must be properly regulated. And ubiquitination is one kind of regulator. Signal transduction is regulated by specific and orchestrated PTM, or post-translational modifications. The post-translational modifications include phosphorylation, simulation, nadylation, and in this class, we will talk about ubiquitination. The post-translational modifications are usually rapid and reversible, thereby ensuring a quick and economical adaptation to microenvironmental changes. Why is it quick? Because for protein synthesis, it will take hours, even several days. But for PTM, it will only take seconds or minutes. For example, when we treat cells with LPS, it will take only less than 1 minute for phosphorylation and ubiquitination to happen. And why is PTM economical? Because for protein synthesis, to terminate signaling transduction, the synthesized protein must be degraded. But for PTM, it's different. In PTM, to terminate the signaling pathway, the modification can be removed, but the proteins remain intact. The proteins are not changed. So it's more economical. The cells don't need to synthesize and degrade proteins, because this will take a lot of time and material. And ubiquitination is a kind of post-translational modification, or PTM, ensuring quick and economical responses to the changing environment. So the proper reaction to the changing environment is very important to maintain health. We take the intestine, for example, because we know in the gastrointestinal tract, there are many commensal bacteria. When there is a problem with the intestine, some bacteria cross the damaged intestinal barrier and enter the intestinal lamina propria. If the cells react correctly, then the innate immune cells, for example, macrophages and some dendritic cells, can kill the invading bacteria. But if they cannot achieve this, then this will trigger a compensatory adaptive immune reaction mediated by T cells. For example, the Th1 cell, Th17 cell, and Th2 cells. And this will cause a big problem because this will lead to the generation of some inflammatory bowel diseases, including ulcerative colitis and Crohn's disease. So ubiquitination serves as a regulator for signal transduction. It ensures that cells react properly to the changing environment. So how does ubiquitination happen? We know that ubiquitination is mediated by ubiquitin. What is ubiquitin? Ubiquitin is a revolutionarily conserved 76 amino acid regulatory protein. It has a molecular weight of 8.6 kilodalton. And this shows the structure of ubiquitin. We can see that ubiquitin is a small protein with 76 amino acids. And this indicates this shows the amino acid sequence of ubiquitin. Ubiquitination, also known as ubiquitination, modifies target proteins by adding one or more ubiquitin molecules. And ubiquitination is catalyzed by a cascade of 3 ubiquitin modifying enzymes. The ubiquitin activating enzymes, or E1. There are 2 E1s in humans. The second one is ubiquitin conjugating enzyme, or called E2. There are about 40 E2s in human. And the third one is the ubiquitin ligase, or called E3. There are around 600 E3s in humans. So how does ubiquitination take place? So in the first step, ubiquitin is activated by an E1 ubiquitin activating enzyme in the presence of ATP. Then the C-terminal glycan of ubiquitin and the cysteine residue of the E1 forms a CO ester bond here. Then ubiquitin is attached to the E1 like this. In the second step of ubiquitination, activated ubiquitin is transferred from E1 to an E2. In the third step, ubiquitin is covalently attached to a protein substrate, and this is coordinated by a specific E3. The E3 ligase determines substrate specificity. If we repeat steps 1, 2, 3, then more than one ubiquitin can be attached to the substrate. We can see from here that a chain of ubiquitin molecules is attached to the substrate. So this is the process of ubiquitination, and this process is mediated sequentially by E1, E2, and E3 enzymes. So we can see from here again that ubiquitin is first activated by the 1E1, and then the activated ubiquitin is transferred to an E2. In the final step, the ubiquitin is attached to a protein substrate by an E3. So if only one monomeric ubiquitin is attached to the substrate, then this is called monoubiquitination, because only one molecule of ubiquitin is attached to the substrate. If some single ubiquitins are attached to the substrate, it's called, for example, this one, on this substrate, there are several single ubiquitin molecules, and this is called multi-monoubiquitination. In addition, ubiquitins can form polyubiquitin chains. For example, here, some ubiquitins gather together to form a chain. And then this ubiquitin chain is attached to the substrate. And then this is called polyubiquitination. So in general, ubiquitination can be divided into monoubiquitination, multi-monoubiquitination, and polyubiquitination. And the type of ubiquitination decides the function of ubiquitination. So in polyubiquitination, the C-terminal glycine residue of one ubiquitin molecule is attached to the methionine residue or one of the 7 lysine residue of another ubiquitin molecule. This forms the isopeptide bond and form a polyubiquitin chain. Then the chain of ubiquitin molecules can be attached to a protein substrate. If the ubiquitins are linked with a methionine residue, then this polyubiquitination is called M1 polyubiquitination or linear ubiquitination. It is called linear ubiquitination because methionine residue is the N-terminal, the first amino acid in ubiquitin, and the glycine is the last amino acid of ubiquitin, so it's called linear ubiquitination. And also depends on the lysine residue. Polyubiquitination can be divided into K6, K11, K27, K29, K33, K48, and K63 polyubiquitination. The K6 means the lysine residue as the 6th position, and this one is as the 11th position. So what is the function of ubiquitination? We know that ubiquitination is a post-translational modification. It is involved in signal transduction. But what is the exact function of ubiquitination? Ubiquitination was initially identified as a mechanism regulating protein degradation. Indeed, the K48 linked ubiquitination signals protein to degradation, and the proteins attached with K48 linked polyubiquitin chains can be degraded via the 26S proteasome. In addition to K48 specific ubiquitination, protein degradation can also be triggered by K6, K11, K27, K29, and K33 linked polyubiquitination. So as I told you before, the type of ubiquitination decides the function of ubiquitination. So we can see from here, K6, K11, K27, K29, K33, and in particularly K48 polyubiquitination signals proton for degradation. In 2004, the Nobel Prize in Chemistry was awarded to the 3 scientists for the discovery of ubiquitin-mediated protein degradation. And this, the protein degradation is mainly mediated by K48 polyubiquitination. However, ubiquitination also regulates non-degradative functions. For example, monoubiquitination. What is monoubiquitination? So it's this is a protein substrate, and if only one molecule of ubiquitin is attached to the protein substrate, then this is called monoubiquitination. And then if some single ubiquitin molecules are attached to this protein substrate, then this is called multi-monoubiquitination. Monoubiquitination and multi-monoubiquitination regulate membrane trafficking, endocytosis, and viral binding. They don't regulate protein degradation. Other ubiquitination, for example, the linear and K63-linked polyubiquitination is critical in intracellular signal transduction. Linear and K63 ubiquitination is a signal for protein activation instead of protein degradation. And we should know that the linear and K63-linked ubiquitination, they are also called they are 2 types of polyubiquitination. In the polyubiquitination, ubiquitins form a chain, and then this chain of ubiquitin is attached to the protein substrate. And ubiquitination has been implicated in the regulation of a broad range of cellular processes, including autophagy, cell cycle regulation, DNA repair, cell death, and immune responses, in particular inflammation. So how can we detect ubiquitination? We know that proteins can be detected by the biochemical method of Western blot. I don't know whether you know this or not, but Western blot can detect a protein. So for example, this protein can be detected by Western blot as a band, only one band, one protein, one band. For example, we take this band as an example. But because ubiquitin is a small protein with 76 kilodalton, then if one ubiquitin is attached to the protein, then the molecular weight of this protein will increase by 7.6 kilodalton. And then it will generate another band. If another ubiquitin is attached, then the band, molecular weight increases again. So the ubiquitination Western blot looks like this, like a ladder. This is, of course, the optimal condition. We can see that the ubiquitinated protein appears to be a ladder in the in Western blot, because with one more ubiquitin, the molecular weight of this protein will increase by 7.6 kilodalton. So this is another example. MDP is a material derived from bacteria. MDP can activate the intracellular receptor NOD2 and also the signaling molecule RIP2. Upon recognition of the bacterial MDP, NOD2 RIP2 signaling is activated. Then RIP2 is ubiquitinated and activated. The ubiquitination signals are required for the activation of RIP2. And we can see from here, the LUBAC and XIAP, they are E3s. They mediate the ubiquitination of RIP2. And from this Western blot result, we can see that RIP2 can be ubiquitinated with K63, K48, K27, and M1 ubiquitination. And different types of ubiquitination can be detected by specific antibodies. So this is a schematic diagram of this of RIP2 polyubiquitination. We can see that different kind of polyubiquitin chains can be attached to RIP2. And K48 polyubiquitination is required for protein degradation, while other polyubiquitination are required for protein activation. Ubiquitination is a reversible process, and it can be counter-regulated by deubiquitinases or deubiquitinating enzymes. We call deubiquitinases DABs for short. So because ubiquitination is a reversible, so it is very economical for signal transduction. And also it's very quick because if the protons are stripped of ubiquitin molecules, then the proton will become normal again. And this process doesn't need new protein synthesis or protein degradation. So ubiquitination can be counter-regulated by deubiquitinases. The deubiquitinases or DABs can remove ubiquitins from protein substrates. They can also cut the ubiquitin chains to release monomeric ubiquitin molecules. In addition, DABs can also release monomeric ubiquitins from the ubiquitin precursor proteins. So ubiquitins are generated as ubiquitin precursor proteins, and in the ubiquitin precursor proteins, the ubiquitins are attached to other proteins or other parts. So DABs can cleave the chain to release monomeric ubiquitin molecules. This will form the free ubiquitin pool, and the free ubiquitins can be used as a raw material for ubiquitination. To date, there are around 100 DABs in humans, and based on structures, the 100 DABs can be categorized into 7 families: the USPs, OTUs, JAMs, UCH, the USAFNs, MINDIs, and SUP1. So this picture shows the DAB family. And DAPs can be divided into 7 subfamilies. The biggest subfamily is the USP subfamily, and it contains around 50 members. The smallest subfamily is the SUP1 subfamily. It has only 1 member called SUP1. Before I concentrated on DAPs from the OTU subfamily. The full name of OTU is ovarian tumor domain ubiquitinases. And different DAPs have different preferences for ubiquitin chains. For example, CYLD is a deubiquitinase, and it has a preference for cleaving K63 polyubiquitin chains. And OTUB1 is a DAP that prefers to cleave K48 polyubiquitin chains. And other, for example, Cezanne likes to cleave K11 polyubiquitin chains. So the function of DAPs is also very diverse. And also DAPs have different subcellular localizations. Some DAPs reside on the plasma membrane, like here, and some DAPs stays in the nucleolus, like USP36 and USP39. And some DAPs stay in the Golgi plug, in the Golgi apparatus. So different DAPs have different subcellular locations. So the process of ubiquitination is catalyzed by E1, E2, and E3 ubiquitin enzymes. In the first step, ubiquitin is activated by 1E1 ubiquitin activating enzyme. In the second step, ubiquitin is transferred to an E2 ubiquitin conjugating enzyme. In the final step, ubiquitin is covalently attached to a protein substrate by an E3 ligase. The K48 ubiquitination is required for protein degradation, but ubiquitination also regulates non-degradative functions. For example, K63-linked polyubiquitin chains give the signal for protein activation, and the K63 polyubiquitination is required for signal transduction. Ubiquitination is a reversible process, and it can be counter-regulated by deubiquitinases or DAPs. DAPs can remove ubiquitin molecules from protein substrates, and this process is called deubiquitination. The deubiquitinase A20 prefers to cleave K63 ubiquitin chains, and the DAP OTUB1 prefers to cleave K48 linked polyubiquitin chains. So after talking about deubiquitinases, now we have to talk about the second concept of neuroinflammation. To study neuroinflammation, I used the model of the neuroinflammatory model of multiple sclerosis. Multiple sclerosis, or MS, is an inflammatory demyelinating disease of the central nervous system. It is an autoimmune disease of the brain. From this picture, we can see that in the brain of the MS patient, there is a lesion, and this is a MS lesion. And that is the reason why this disease is called multiple sclerosis, because there are many sites of sclerosis in the brain. MS is pathologically characterized by perivascular infiltration of inflammatory cells, demyelination, and axonal damage in the brain and spinal cord. In the brain, there are many neurons. We all know that neurons play important roles, and the axon of a neuron is covered by myelin sheaths, the yellow myelin sheaths. The myelin sheath serves as the insulation cover for the axon. It acts like the plastic cover for the cable. In multiple sclerosis, the immune cells, particularly T cells and B cells, they recognize the myelin as foreign material. So they attack the myelin sheath. Then the myelin sheath is damaged, like the damaged cable. So we all know that if the cable is damaged like this, we cannot use it because it's not functional anymore. And an attacked neuron also like this. So they cannot, a neuron with damaged myelin sheath can also not function properly. So the MS patients show some symptoms, for example, visual disturbances, paresthesia, paresis, and ataxia. MS is the leading cause of paralysis in young adults resulting from neuroinflammation in Western countries. And in European countries and Australia, there are many MS patients. So to study MS, we use animal model of experimental autoimmune encephalomyelitis, or EAE. This is an animal model for MS. To induce EAE, we inject peptide into the mouse. This peptide mimics the myelin sheath in the central nervous system. If the mouse receives the peptide, then T cells and other immune cells will recognize this injected peptide and then attack myelin, because myelin is similar to this peptide. So after 10 days, mice will develop symptoms. For example, the paralysis of hind limbs and tails. Previously, I studied 2 deubiquitinases in EAE. The first studied deubiquitinase is A20. A20, also called tumor necrosis factor alpha induced protein 3. It is a DAB encoded by the TNF-AIP3 gene. Single nucleotide polymorphisms in or close to the human TNF-AIP3 gene is associated with many autoimmune diseases like psoriasis, rheumatoid arthritis, type 1 diabetes, systemic lupus erythematosus, and multiple sclerosis. Right, multiple sclerosis is a neuroinflammatory disease that that we will talk we focus on today. So this is a structure of A20. It has an N-terminal OTU domain. This domain confers A20 the DAB activity or the ubiquitinase activity. A20 also has several zinc finger domains. The zinc finger domain 4 confers A20 the E3 ubiquitin ligase activity. So A20 is a very special one. It has both DAB activity and E3 ligase activity. That means A20 can not only add ubiquitination, but it can also remove ubiquitination. So from this picture, we can see that upon activation with stimulation with the TNF, it's a pro-inflammatory cytokine, or LPS. LPS is derived from bacteria. Then the intracellular NF-kappa B signaling pathway is activated. And A20 can inhibit the activation of NF-kappa B signaling. In the last slide, I said that A20 has both DAB activity and E3 ligase activity. We can see from here, A20, this one is A20. A20 can remove K63 polyubiquitin chains from RIP1, and it can also add K48 polyubiquitin chains to RIP1. So A20 can not only remove ubiquitin, but can also add new ubiquitin to the protein substrate. Before I studied the function of A20 in EAE, as I talked before, EAE is a model used to study the human autoimmune disease, MS. And in the paper that I published, I found that A20 inhibits EAE by inhibiting the NF-kappa B signaling in astrocytes. And this paper was published in 2013 in Acta Neuropathologica. And this result is cited by Nature Reviews Neuroscience as a very good journal in neuroscience. So in the second project, I studied the function of another ubiquitinase called OTUB1. So unlike A20, which has a preference for cleaving K63 polyubiquitin chains, OTUB1 likes to cleave K48 polyubiquitin chains. OTUB1 has a N-terminal ubiquitin binding domain and a C-terminal OTU domain. The C91 residue or the cysteine 91 residue is required for the the ubiquitous activity of OTUB1. OTUB1 is involved in the regulation of many signaling pathways by deubiquitinating and stabilizing signaling molecules because it can stabilize signaling molecules because it cleaves K48 polyubiquitin chains. We learned before that K48 polyubiquitin chains are necessary for protein degradation. If OTUB1 removes K48 polyubiquitination, then the protein substrate can escape protein degradation. So before I found that in MS brains, OTUB1 is highly expressed in astrocytes of MS patients. So the red cells are astrocytes. And OTUB1 expression levels are elevated in astrocytes during MS. So these results indicate that OTUB1 plays a role in multiple sclerosis or MS. So to study the function of OTUB1 in multiple sclerosis, we will use animal models because we cannot do experiments directly on humans. So to study the function of OTUB1, we want to generate the OTUB1 deficient or knockout mouse. To generate the OTUB1 knockout mouse, we first generated OTUB1 flox mouse. In this mouse, exons of exon 2 and 3 of OTUB1 are flanked by LOXP sites to generate the OTUB1 flox mice. If you haven't done some experiments with animals, maybe it's difficult for you to understand. But you should know that in OTUB1 flox mice, the OTUB1 gene is modified. So this gene can be deleted later. Then we crossed OTUB1 flox mouse with some Cree mice. And with this technology, we can specifically delete OTUB1 in neuroectodermal cells, including astrocytes, neurons, and oligodendrocytes. So in this type of OTUB1 knockout mouse, OTUB1 is only deleted in these cells, in the 3 cell populations. But in other organs and cells, OTUB1 remains intact. This is called tissue-specific OTUB1 knockout mouse. But unfortunately, the mouse, the mouse deficient of OTUB1 in these 3 cell populations can not survive due to embryonic lethality. Then we generated another OTUB1 knockout mice. In this mouse, OTUB1 was specifically deleted in astrocytes. So we generated one mouse strain with OTUB1 deficiency only in astrocytes, only in these astrocytes. These mice were born in a normal Mendelian ratio and reached adulthood normally. So they didn't show any abnormalities. They grew normally. But upon EAE induction, So we induced the EAE in these mice, and we found that OTB1 as an astrocyte-specific OTB1 knockout mice developed more severe EAE. So the disease, the EAE disease in the knockout OTB1 specific knockout mice is more severe. Then we analyzed the infiltrating immune cells in the spinal cord during EAE, because in mice, in EAE mice, many immune cells invade the spinal cord to destroy myelin there. And we found that in astrocyte-specific OTB1 knockout mice, there were more infiltrating CD4 T cells in the spinal cord. We also analyzed the subsets of invading T cells, and we found that OTB1 knockout mice had more disease promoting CD4 T cell subsets. We have to know that maybe it's difficult for you to understand now, but you have to keep in mind that CD4 T cells are bad for MS and EAE. So more CD4 T cells means that more severe disease. So upon EAE induction or mock immunization, myelin reactive T cells are primed in peripheral lymphatic organs, and some T cells can invade the subarachnoid perivascular space through leptomeningeal vessels or choroid plexus. In this area, the subarachnoid perivascular space, T cells are reactivated by antigen-presenting cells, including dendritic cells and macrophages. As a result, CD4 T cells undergo clonal expansion and produce a lot of pro-inflammatory cytokines, including TNF, interferon gamma, and interleukin-17. These cytokines activate or stimulate adjacent CNS resident cells, particularly astrocytes. Activated astrocytes produce leukocyte-recruiting chemokines, and the chemokines can recruit a large group of leukocytes to the central nervous system. This is associated with demyelination. We can see from here that the myelin sheath is damaged by the infiltrating T cells and other immune cells. And this is associated with EAE onset. So we can see from here that astrocytes contribute to EAE progression and onset by producing pro-inflammatory chemokines. Because OTB1 deficient or astrocyte-specific OTB1 knockout mice developed more severe EAE than control mice, is it possible that this is caused by the increased chemical production by OTB1 deficient astrocytes? To confirm this, we isolated astrocytes from EAE mice. and then analyzed chemical production. Indeed, the astrocytes from the OTB1 deficient or knockout mice produced more chemokines than do control astrocytes. And later we also confirmed that OTB1 inhibits interferon gamma-induced astrocyte activation. And OTB1 doesn't play a role in astrocyte activation mediated by IL-17 or TNF. IL-17, TNF, and interferon gamma are fingerprint cytokines secreted by autoimmune T cells during MS and EAE. And from this result, we can see that OTB1 only inhibits interferon gamma induced astrocyte activation, because we can see this is a good example here, because the black bar means OTB1 deficient or knockout astrocytes. The white bar indicates the normal astrocytes. We can see that after the deletion of OTB1, astrocytes produced more CXCL10. CXCL10 is one chemokine upon stimulation with interferon gamma. So no OTB1, more production of CXCL10. So OTB1 inhibits interferon gamma-induced chemokine production in astrocytes. And later, with Western blot, I also found that OTB1 inhibited interferon gamma-induced intracellular signaling pathways, the JAK-STAT signaling pathways. And so we can split this membrane into two halves. This side is control astrocytes, and this side is OTB1 knockout. We can see that from this membrane, we can see that OTB1 is deleted in OTB1 knockout astrocytes. And upon interferon gamma stimulation, the phosphorylation of STAT1 is increased in the knockout cells. Especially, for example, here, this band is very strong and this band is weak compared with this band. So we can see from here that OTB1 deficiency increased signal transduction in astrocytes upon interferon gamma stimulation. And to understand why OTB1 inhibited interferon gamma signaling pathway, I did another experiment. In this experiment, I found that OTB1 interacted directly with SOX1. SOX1 is a protein, is an inhibitor for interferon gamma-induced signaling pathways. And also OTB1 can cleave K48 polyubiquitin chains from SOX1. And later I also found that the N-terminal ubiquitin binding domain of OTB1 is required for cleaving K48 polyubiquitin chains from SOX1. And this is with this result. because K48 polyubiquitination regulates protein degradation, OTUB1 can stabilize SOX1 because OTUB1 cleaves the degradation promoting P48 polyubiquitination. And for example, we can see from here, the degradation of SOX1 is accelerated in OTUB1 deficient cells. And also with one experiment, I show that SOX1 knockdown blunts the difference in gene expression between OTUB1 sufficient and OTUB1 deficient astrocytes. That means OTUB1 inhibits interferon gamma-induced chemical production in astrocytes by directly targeting SOX1. So in this project, I found that OTUB1 ameliorates EAE by inhibiting interferon gamma-induced chemical production in astrocytes via stabilizing SOX1. And this paper was published in 2019 in EMBO journal. And also this result was also published in Brain Behavior and Immunity in 2019. So this paper and also the A20 paper can be found on internet. If you are interested, you can have a look. So thank you for your attention. If you have some questions, you can write to me. This is my email address. And thank you for your listening. Thank you.
You're welcome, sir.
Thank you, sir.
Okay, so do you have any questions? Do you have any questions?
No, sir.
Okay, so I hope you enjoy the class. And then if you have no questions, so what?
Thank you.
Thank you so much.
Okay. Yeah. Thank you for your listening. And so if you have some questions or if you want to contact me, you can write to me. I have this email, and I can write. So now I'm interested in delineating the function of ubiquitinases in inflammatory diseases. So we used some disease models like the EAE model. We also have some stroke model, stroke, the burn, some artery problem. And also we have some colitis model. And if you are interested in research in these fields, you can maybe, if you have the opportunity to come here, and I think it's a good opportunity to study this project. So if you have no more questions, so we finished the class. And thank you again for listening, and hope to see you again. Okay, so bye.
Bye.
Bye, sir.
Bye, sir. Bye.
Goodbye, sir.
Bye.
Good day.
Have a nice
Bye.
day. Have a
Yeah,
nice
the same to you. Yeah.
Have a nice day, sir.
Okay, bye.